Thank you, Tom, for the invitation and presentation. Can you all see my presentation?
Yes.
Good?
Yeah, we
Okay,
can.
thanks. So my presentation is entitled Investigating the Brain Correlates of Chronic and Acute Stress in Pig Models. And I'm going to present you some results combining behavioral approaches with functional brain imaging in the pig model. So I have no conflict of interest to declare, only a link of interest with FODE, which is a French SME specialized in olfactory sciences for animal, but also human nutrition, health and welfare, because part of the scientific data presented here today was obtained in collaboration with them. So I will be very quick and brief on this general slide, because you all know that stress can be considered as a significant burden in our modern societies, and some authors even describe stress as the health epidemic of the 21st century. So of course it concerns humans, but also animals. So you all know the definitions, or at least one definition of stress, which consists in the responses of organism subjected to pressures, threats, or constraints, those being established to recover homeostasis, but also well-being. So we have to differentiate stress and stresses, the events or stimuli that cause stress. And we also have to distinguish between physiological stressors, such as hypothermia, dehydration, or hypoxia, and psychological or psychosociological stresses, such as social conflict, rejection, or social isolation. So to cope with stress, acute stress or chronic stress, the individuals can express physiological adjustments, but also coping behaviors. And as you explained in your introduction, Tom, acute stress can become chronic stress when the events are repeated, unpredictable, or uncontrolled. And we all know that chronic psychosocial stress makes the bed of depression, but also anxiety, as well as a long list of different types of disorders, so somatic disorders, but also cognitive, mood disorders, and also appetite disorders. And part of my research, most of my research is dealing with food disorders, eating disorders, and the influence of stress is well known and well documented in animals, but also in humans. So today I will talk about the research we've been performing in the pig model. I've been doing research in the pig model for more than 15 years now, and this species is a very interesting one for behavioral and neurosciences and nutrition studies. Pigs are an omnivorous species with a digestive system and a brain which are very close to those of humans, which means that we can perform brain imaging with similar machines and modalities in humans and in animals, which is very interesting for preclinical and translational science. So when doing research with pig, we can use conventional pigs, but also miniature pigs. Conventional pigs are the meat-producing pigs, so they are quickly growing and widely available, but the downfall is that they are very big, and at the adult age, they can weigh more than 250 or 350 kilograms. So sometimes miniature pigs are a very good asset to do research, because they are easy to handle. They can weigh 50 to 80 kilograms at the adult age, but they are not obese. So it's a very good model. But today I will present only research that have been performed in conventional pigs. So in our department, we have implemented many types of behavioral tests in pig models to investigate, for example, food preferences, but also cognitive abilities, food-rewarded cognitive abilities with the whole board test, which has been adapted from mice and rodent tests. We have also developed operant conditioning cages to investigate food motivation and choices, but also some tests to investigate, for example, a cognitive conflict between food motivation and anxiety or fear induced by a novel object. We have also developed other types of tests to investigate emotional reactivity to a novel area or spaces, such as the open field, complex maze tests, but also this one I will present it later, the novel moving object test to investigate the fear reactions toward a frightening stimulus. We've been using also the contention test in a Pavlov stand, which is useful to investigate perseverance and resignation. So I would like also to introduce how we are doing brain imaging, functional brain imaging in pigs. So when the brain is activated, the neurons need oxygen and glucose. So meaning that when neurons are activated, you can see locally an increase in terms of vascularization, but also an increased oxygen and glucose consumption. And in France, in Rennes, we are equipped with different types of machines to investigate different indicators or markers of brain activity. For example, we can use functional magnetic resonance imaging to investigate the hemodynamic response in the brain, the Bolt signals. We can use single photon emission computed tomography to investigate cerebral blood flow, but also positron emission tomography to investigate brain glucose metabolism. Of course, all of these modalities and indicators are correlated with each other. So when we do brain imaging in pigs, we must anesthetize them. So you will ask me, how do you investigate the brain responses if the pigs are sleeping anesthetized? Well, we developed some tools, some computer-controlled automats to stimulate the animals, for example, with an odor and a taste, stimulation of the tongue or in the snout, to stimulate the brain and investigate the brain responses. And even if the animal is anesthetized, the brain is still able to respond to these stimulations. So we can invent different types of stimulation. For example, here we have a combination between a duodenal stimulation with a nutrient, for example, and an oral stimulation. And when the pig is equipped, it has a tube going to its snout, another tube going to its tongue, and we can stimulate the animal with different types of flavors, odors, taste. But we've also performed auditory stimulation and visual stimulations. So I will not present these today. But for example, we investigated the brain reactions to different types of vocalizations, positive vocalization, reunion with the mother, for example, or negative vocalizations, for example, when animals are castrated or they are separated from their mother. So speaking now about the chronic stress, psychosocial stress model in the pig, we wanted to describe a pig model of chronic psychosocial stress using social isolation. So we had a control group of pigs who were housed in pairs in an enriched environments with churros and toys, and we've decided to propose two different groups, experimental groups. One group was the SC minus, the minus stress. Pigs were isolated, so there was a social isolation, and the environment was impoverished. There was no toys, for example, or no stray. And another group was called SC+, and in this group we've added another stressing stimulus, which was stressing lights and sounds at different types or at different hours of the day. So this was an unpredictable stressing stimulus. And in both these groups, we've treated half of the animals with fluoxetine, which is an antidepressant. So what we did is different types of behavioral observations in the housing pen of these pigs, as well as biological sampling, including saliva for cortisol assessment, blood samples, and feces samples for investigating microbiota. So we've been doing a behavioral test, and then before the functional magnetic resonance imaging. So the first result I would like to show you were those obtained during the contention test. And what we observed is that only the SC+, so these animals that were not treated with fluoxetine, showed a decreased perseverance index, which means that they resonating very quickly when they were confronted with this type of test, contention test. And this was correlated with a higher secretion of cortisol only in this group. And this cortisol secretion was completely countered by the fluoxetine treatment. So for the next slide of my presentation, I will only talk about these two groups, which were really the most interesting groups, the control group and the unpredictable stress model. So during the housing period, we've explored the different behaviors expressed by the animals in their cages. And we've been doing a principal component analysis to compare the different dimensions of the animal behavior. And what we can observe here is that the non-stressed animals that were housed in pairs and with an enriched and predictable environment, they expressed more playing and exploration behavior, more silent. In comparison, the stressed animal in the unpredictable environment were expressing more grunting, squealing. They were looking at the human with anxiety, for example, as well as more stereotypic behaviors. So these animals were subjected to chronic psychosocial stress condition displayed significant differences in terms of behavior, including depressive-like behaviors. Now, when we've done the functional magnetic resonance imaging, our hypothesis was that reaction or responses to new food orders might be different in stressed animals. So here you can observe MRI, fMRI acquisition, ball responses in the brain when animals were exposed to new food orders. And you can see here the frontal cortex, prefrontal cortex, the cerebellum here, so it's an horizontal view. And you can see activated areas in red here in stressed animals and deactivated areas in blue. So the induction of the psychosocial stress modified the brain responses to novel food orders, notably in the hippocampus here, which is involved in learning but also emotions, and prefrontal cortex, which is involved in hedonism and executive control, notably over food control. What's interesting is the fact that during MRI, we've also performed magnetic resonance brain spectroscopy to investigate a marker of neuronal density. And we've shown that stressed animals here expressed a decreased ratio between these two markers, which can be interpreted as a lower neuronal density in the hippocampus. So at the end of the experiment, we've done immunohistochemical analysis. And what we've shown in these stressed animals in red here, compared to the control animals in green, is that stressed animals showed less cell proliferation in the hippocampus, even though there was no difference in terms of cell differentiation. And interestingly, and in comparison to what Marco told us in fishes, we also observed a decreased expression of the serotonergic receptor 5-HT1A-R, so in the hippocampus in stressed animals, which was significant. Apart from these differences in terms of behavior, but also brain responses, we've also performed different types of exploration at the gut and microbiota axis. For example, we've shown that there was a dysregulation of the glucose homeostasis in stressed animal, which was not treated by fluoxetine here. We've also observed differences in terms of the functional gut barrier, with less crypt goblet cells in the intestinal barrier, but also an increased gut permeability in stressed animal. That means that potentially pro-inflammatory compounds from the microbiota could go into the plasma and potentially to the brain to induce consequences at the gut-brain axis level. And finally, in terms of gut microbiota activity, in terms of short-chain fatty acids productions, we also observed a decreased fermentation activity of the gut microbiota in stressed animal, which was only partially countered by fluoxetine. So all these results show that there was a modulation of microbiota gut-brain axis by chronic psychosocial stress, with only positive effects of fluoxetine on some parameters. So after that, we wanted to investigate how we could modulate these responses to chronic stress, and that's where we've collaborated with Foley SME. So in traditional medicine, but also modern aromatherapy and phytotherapy, a lot of vegetal plant extracts are used to have an impact on health. For example, hot spices extracts, but also citrus extracts, which have been described for for their antioxidant, anti-inflammatory, but also antimicrobial activity. And in the context of mood disorders or stress disorders, some of these functional ingredients have demonstrated improvements in terms of mood. They have been described to modulate brain activity and monoamine systems, with some effects on food intake regulation, but also improvements in terms of metabolic disorders, gut barrier protection, and gut microbiota modulation. So that's why we wanted to test some of these plant extracts in the context of our pig model of chronic stress. So what we did was to perform an experiment or set a different experiment with this unpredictable stress model of pig. So I will present here two different experiments, independent experiments, which are a bit different from each one another, but they have similarities. So the first experiment used an ingredient that was conceived by Fode, which was a combination of curcuminoids, piperines, gingerol, shogal, capsaicin seeds, so hot spices. So we've been doing behavioral tests, for example, the open field test and the novelty suppressed feeding test before doing brain imaging explorations. And in another protocol, we've done similar exploration, but with another ingredient with citrus sinensis extracts. And we did not repeat these tests, but did the contention test, for which we've observed positive results in the psychosocial stress. So, looking at the results from the first experiment on the food ingredients mixing hot spices, what we've observed was a decreased number of escape in the open field test in mini-pigs that were treated with these functional ingredients. And during the novelty suppressed feeding test, where the animal has to fight against its fear of the novel object test to feed in the trough, well, the latency to reach the feed was decreased in animals treated with the ingredient, which means that the animals were probably less stressed or less anxious when chronically treated with the ingredients mixing hot spices. So when we look at the basal brain metabolism of these animals, so meaning that there is no stimulation in this context, but only the basal activity of the brain. So we've observed differences between groups, control groups, and treated groups. For example, an increased activity in the olfactory bulbs in animals that received chronically the ingredient with hot spices. And we've also observed in these animals a decreased activity in some regions, for example, the dorsolateral prefrontal cortex, but also in the striatum, the caudate nucleus here, and the hippocampus, and also in the thalamus. And most interestingly were the immunohistochemical and biomolecular results we've obtained in these animals, because treated animals showed higher expression of BDNF, which is a regulator of cellular processes and synaptic plasticity, so which is very important for neuronal plasticity. And we've also observed an increased expression of a 5-HT1AR receptor, so the serotoninergic receptor was increased in animals that received the functional food ingredient. And remember, these receptors were down-regulated in the context of chronic stress. So, to sum up the results, the functional food ingredients had modulatory effects on behavior with anxiolytic properties, but also on brain functioning and possibly on neuroplasticity and neurotransmission. Unfortunately, we did not observe any results in terms of metabolic, gastrointestinal, and microbiota parameters in this study. So the second protocol used another ingredient with citrus sinensis extracts. And in this experiment, the question was a bit different. We wanted to know whether chronically stressed animals that were treated or not with the ingredient would react differently to an acute stress. And the acute stress that we've been performing during the fMRI, the imaging session, was pharmacological stress, consisted in the injection, venous injection of synactin, which is an agonist of ACTH. And this injection of synactin induced in 30 minutes a significant increase of cortisol secretion. So that was a very interesting way to induce an acute physiological stress in these animals. And we've compared the responses of control animals and animals that were chronically exposed to the ingredient with citrus extracts. First, what we've been describing is that injection of synactin, so this pharmacological acute stress, provokes a massive activation in the brain. So the pharmacological acute stress increased brain activity in many regions. What is very interesting in this study is that in the animals that received chronically before the brain imaging session this ingredient, well, these responses to stress were down-regulated. So supplementation with the ingredient limited the brain reactivity to pharmacological stress, which was very interesting. Unfortunately, once again, we did not observe any effect on behavior, gut permeability, or microbiota, which was really deceitful, but only these results on brain are already very interesting. And I would like to finish by a third experiment that we've been performing. And in this context, we used another food ingredient combining aromatic fatty acids, vanillin, threonine, and diacetyl, which has been used in the literature in humans, but also in pig nutrition. And what we've done here is that we did not expose the animals to chronic stress. So all the animals were housed in good condition. They were housed in pairs with an enriched environment with toys. So and they were not stressed by lights or sounds. So what we did in these animals is that some of them were control animals, and some of the animals received the ingredients. And all the animals were subjected to feed choices, a novel object test, and the contention test, for which we have observed significant results in psychosocial stress. And once again, we've done fMRI in all the animals, 12 animals per groups. So the novel object test consists in the introduction of a moving object after a period of adaptation to the open field. So a balloon is going down, it's moving, and simultaneously there are lights and sounds, which is really frightening for the pigs. And our hypothesis was that exposure to this ingredient would reduce the anxious or stress responses of pigs. Unfortunately, we observed the contrary. Pigs that were receiving the ingredient showed more emotional reactivity when confronted to the novel object test. For example, they spend more time in the peripheral zone here and less time in the balloon zone. And when we looked at the latency to reach the balloon, it was increased in animals exposed to the ingredient. So that was not the most interesting part of the study, because that was not an agreement with our hypothesis. So here are the results from the brain imaging, which are much more interesting. So what we observed is that previous exposure to the functional food ingredient modified the brain responses to the acute pharmacological stress in frontostriatal and limbic circuits. And notably, the pharmacological stress provoked brain activation in the amygdala, prefrontal cortex, and thalamic region, which was current with Menson et al., the previous paper I showed you. But some of these activation were again attenuated in animals that were habituated to the functional ingredient. So this is very interesting, because once again, the stress reactions, the brain stress reactions, were down-regulated in exposed animals to the ingredient. Another interesting result was that animals that were habituated to the functional ingredients also showed more activation in the striatum, here in the caudate, for example, or putamen, which are involved in pleasure and motivation, but also in the peripheral area, which is part of the limbic system and very important for olfaction and emotions. So in conclusion, acute physiological stress, but also chronic psychosocial stress, can induce specific brain responses in the pig's brain, especially in the methylamine pathway. So we've described that there are hippocampal plasticity differences and serotonergic system alteration by chronic psychosocial stress, and that these brain anomalies are associated with altered responses to novel food orders, more stereotypic habit behavior, less playing and exploration behaviors, and increased anxiously perceived behaviors. And finally, these brain anomalies are associated with modifications at the microbiota and gut levels. In terms of research perspectives, we could explore and compare the brain responses to acute versus chronic stress, compare also different stresses, because here we only had one model of psychosocial stress, chronic stress. The switch also between adaptive to maladaptive responses, but also the communication pathways at the microbiota gut-brain level, as well as the impact of stress on cognition and behavior. And my last slide recapitulates all the results we've obtained in the pig model and different experiments, showing that the pig model is a convincing model for chronic psychosocial stress. So we've shown that fluoxetine can have an effect on behavior, but also on gut-brain axis. But the results we've obtained in the functional ingredient, even if they produced very interesting results at the brain level and some of them at the behavioral level, we've not managed to demonstrate that they could alleviate the stress effects at the gut-brain, at the gut axis and microbiota levels. So in terms of perspectives, we would like to work on the dose, the formulation, and modes of administration of these ingredients, because all of these ingredients were consumed, ingested, so perhaps there are other modes of administration that would need to be explored. The effects on the behavior must be explored with other tests, because those we've used did not produce significant results in all projects. And finally, we would like to explore the pathways of communication, the mechanisms underlying these effects. For example, I talked about the olfaction versus ingestion action modes, but we could also imagine blockades of pathways, serotoninergic or GABAergic pathways. And because some of the ingredients we've been using are a mix of different molecules, it would be also very interesting to investigate the interactions between these different molecules. Thank you very much for your attention.
Thank you very much, David. As before, stick up your hand or put a question in the chat. I will monitor both. In the meantime, maybe I can again start the question while other people. Oh, no, we don't need to. Block has a question. Hold on. I need to allow you to actually talk. Oh, no, sorry, click the wrong button. There you go.
Thank you, David. Thank you, Tom. Beautiful talk. David, I have kind of a question out of left field, because I know you spoke about the mini pig model versus kind of the traditional pig model. I'm wondering, are there particular breeds of pig that are perhaps more resistant to some of these stresses? You know, I think from an African perspective, we've always been interested in trying to find farm animals that are more hardy to ecological stresses. Are there anything that you think at this point in time, or that we could kind of explore this, you know, within the pig framework?
Well, all the results I've been presenting today were obtained in conventional pigs that we've been breeding in our station in Rennes. So they are large white landrace pigs used for pig production. So we did not compare the stress responses between different breeds. What I've been doing a few years ago was to compare different breeds of miniature pigs. So we've been comparing Göttingen mini pigs, Pitman-Moore mini pigs, Yucatan mini pigs, and Vietnamese mini pigs. And even though we did not do the types of experiments on chronic or acute stress like I've presented today, there were obvious differences in terms of behavior and emotional reactivity between miniature pigs breeds. So you're right, there are probably some differences between breeds, even between conventional breeds. So I'm not aware of that literature, but I'm quite sure that there are differences in terms of sensitivity to stress or stresses. For example, hypothermia, some pig breeds are more sensitive to hypothermia and are not very resistant to cold temperature or hot temperatures. So depending on the stresses, psychosocial stresses or physical and environmental stresses, you can observe many differences between breeds.
Thank you, David. And it wasn't a take, stab at your work, but it's rather the idea of, how could we push the farming industry to think a little bit about, breeds that would be a little more resistant to kind of the stresses that we're trying to aim at from an animal welfare point of view.
So I did not get your question. Could you repeat, please?
I was saying it was more directed, the question's directed at kind of the farming industry, what are the breeds that would perhaps be most suited,
Well,
from the meat industry perspective?
First, I think we would need to describe an easy implementable type of test to investigate different types of stresses, either environmental or psychosocial stresses, because it's very constraining to do these behavioral experiments. So I guess for physical stresses like temperature or hygrometry or pig density, it can be done quite easily. But as soon as you want to investigate behavioral stresses, social stresses, it becomes more tricky to investigate and compare. But yeah, perhaps some studies are needed to look for new breeds or mix of breeds that would be more resistant to different types of stresses. I'm not aware of that specific work. So I have some colleagues in Rennes that are working specifically on animal welfare, pig welfare, and breeding systems. So I'm sure some of them will be interested by these questions. But since I'm more interested in the human nutrition welfare, I'm not a big specialist, a huge specialist of this literature.
All right. I have a question actually, and that's coming from a little bit, I know, and there's not much there about the human literature in particular. So fluoxetine is one of those drugs that you need to take for quite a long time before it has effects for people with depression. How long were your treatments with fluoxetine, and how long do you need to pre-treat before you see these effects in the pigs? And then a similar question about your active ingredients, your spice extracts, your citrus extracts.
Yeah,
how long do you need to treat with those before you see anything?
That's a very interesting question, and we were wondering that when we've conceived our protocol, and we wanted to know if the sensitivity of pigs would be similar to that of human, because you're right, you need to treat for quite a long time with fluoxetine before observing some positive outcomes in the human. So fluoxetine was provided to the animals from habituation, so during all the experiment, but there was no pre-treatment with fluoxetine. So we can also guess that some effects might have been much more potent if we would have been treating the animals before or prolonged the period of treatments. So they were treated only a few weeks during the stress events, meaning that there was no pre-treatment. And perhaps if the treatment of fluoxetine was performed before the occurrence of a chronic stress or during a longer period, we might have observed different types of responses. But the same question can be asked for the functional ingredients, because there was no pre-treatment with the functional ingredients, and the time course of treatment was similar to that of fluoxetine. So we might imagine that even for functional food ingredients, some molecules might need several weeks before being effective in pigs. So that's an interesting question, but I don't have the answer right now.
Always easy to ask the question than to give the answers. Thank you very much. Right, if there are no further questions for David, I'll say thank you very much, David, for your fascinating presentation. And we will move on to the next speaker, who is Loretta Medina, who is going to tell us about comparing telencephalic centers that regulate stress in sauropsids